Hello students, we are going to start a new chapter, the chapter 4, the animal kingdom, and I have divided this chapter into few segments, and the first part that we are going to discuss about the base of the classification of the animals. Okay, so the first we can move on to the slide. Here we are going to discuss about the base of the classification. For the classification of animals, there are some of the common features that is adapted, and these common features are like the cellular arrangement, the symmetry of the body, the presence of absence of the coelom, then the specific features of the digestive tract, reproductive tract as well as the circulatory system. And based on all these criteria, that is the arrangement of cells, the symmetry of the body, the presence or absence of the coelom, the features of the digestive, circulatory as well as the reproductive system, and following this criteria or based on these common fundamental features, the animals are classified. And first one is the level of organization. In the level of organization, the first one is a cellular level of organization. That is a base level of organization. Here the name itself, that is a cellular level, that is a cell level, that is cells are arranged as a loose clusters or loose aggregates of the cells that is mainly present in the sponges, that is porifera. Okay, there is a tissue level of organization is the next one. Here the cells are arranged in a more complex forms. That is, initially the cellular level only the cells will be there and they are actually arranged as a group of aggregates of the cells. And in the tissue level, these cells, they are actually arranged in a more complex forms, like they are performing the same function. The cells that perform the same function forms the tissues. Okay, that is, in the tissue level of organization is actually little bit higher level. That is coming to the next level, the tissue level. That is, cell performing the same function forms the tissue. The mainly seen in the cylindrites. Next is the organ level of organization. In the organ level of organization, that is the next higher level. Here the tissues, the cells that perform the same functions form the tissue. The tissues that are grouped together to form a particular organ, and these single organs that performs a specific function or particular functions they are performing. Okay, that is, in the organ level of organization, tissue group together to form the specific organs and each organ performs the specific functions. Examples, that is a platyhelminthes. And next is the next level is the organ system level. Here the organs, not the single organ, the group of organs or few organs, they coordinated or they join together to perform a certain physiological functions. That is example, annelids, arthropods, mollusks, echinoderms and chordates. That is, these are the level of organization. First one is the cellular level of organization, second one is the tissue level, third one is the organ level, and the fourth one is the organ system level of organization. In the cellular level, the name itself indicates that it's at the cell basic level. That is, a group of cells perform some functions. Their cells are grouped together, and that is in the sponges. Next is the tissue level. Here it's actually little complex forms of the cells, that is, cells are arranged in little bit complex forms, that is, in that cases the cells that perform the same function form the tissues. Example, cylindrites. And next is the organ system level, the tissue that are grouped together to form the specific cells, and is not the specific cells, specific organs, and each of these organs performs the particular functions. Example, platyhelminthes. Next, the organ system level of organization. In the organ system level of organization, the tissue forms the organs, right? Organ level. Organ system level means these organs, the group of organs together to form a certain physiological functions. They act together and forms the functioning some of the physiological activities in our body, in the bodies of the organisms of the animals. Example, annelids, arthropods, mollusks, echinoderms and chordates. Next is based on the digestive system. The digestive system, the presence or absence of digestive system, or whether they have the single opening or the have the multiple opening is there. The digestive system is classified as an incomplete digestive system and a complete digestive system. In the case of incomplete digestive system, the system has only one single opening to outside of the body that serves as both mouth and anus seen in the platyhelminthes. Okay. In the case of liver fluke and all, we can see that as a platyhelminthes, they have only single opening at the top. That is actually functioning as a both mouth and anus. Next is a complete digestive system. In the complete digestive system has a two opening, mouth and anus will be there. They individually performs each of their function. Mouth is performing the input function and anus is performing the output functions. So that is a digestive system based on the presence of the opening, that is a single opening or the multiple opening. The digestive system is actually incomplete and complete. In the case of incomplete, there will be only one opening that is serving as or functioning as a both anus and the mouth. And the complete digestive system, two openings will be there. One is the anus and another is the mouth. Next is the circulatory system. In the case of circulatory system, there are two types of circulatory system, that is open circulatory system and the closed circulatory system. In the open circulatory system, in which cells and tissue directly receives the blood pumping out of the heart. That is open circulatory system. In the open circulatory system, the cells and tissue directly receive the blood pumping out of the heart. Okay. And in the closed circulatory system, here there is a network of, that is arteries, veins and capillaries will be there, and blood is circulated through this system. This is a closed. There will be arteries is there, capillaries is there, veins are there. They actually club together or join together and forms a network, and the blood is actually circulating through this. That is the difference between the open and closed. Open means, see, you can see in the picture, you can see here, see in this picture you can see there is a hemocoel is formed. That is, from the artery, from the veins, the blood is collected in one area. That is, receive the blood pumping out of the heart directly to the cells and tissues. In the case of closed, you can see there is a closed system, and this blood is circulated through the artery, capillaries and vein. Next is a symmetry. And in the case of symmetry, there will be three types of symmetry, that is asymmetrical, radial symmetry and bilateral symmetry. In the case of asymmetry, there is no line of symmetry in the body. That is, any plane that passes through the center does not divide them into equal halves. That is asymmetry, no symmetry. That is means the asymmetry. Okay, example sponges. If you try to divide the sponges into half, but there you cannot see, there will not be any plane or any line that can divide these organisms into two halves, equal halves. Okay, that is meant by asymmetry. And in the case of radial symmetry, that means any plane passing through the center divides the body of these organisms into two symmetrical, that means identical halves. Example, coelenterates, ctenophores, echinoderms. Okay, that is, in the case of radial symmetry, any plane passing through the center divides these organisms body into two identical halves or symmetrical halves or similar halves. Okay, that is called radial symmetry. Coelenterates, ctenophores and echinoderms. Next is a bilateral symmetry. Bilateral symmetry means there is only a single line or only one plane that divide the body of organisms into equal or identical left and right halves. Example, annelids, arthropods, etc. That is, bilateral symmetry means one line or any one plane that divides the body of organisms into identical left and right half. Example, annelids and arthropods. And in the case of echinoderms, they exhibit radial as well as a bilateral symmetry at different stages of their life. And they are passing actually through the different stages in the lives, and based on the stages, they exhibit either radial or the bilateral. That means they exhibit both the radial as well as the bilateral symmetry. That is echinoderm. That is an exceptional case. This is a picture that shows a symmetry. That is, first one is a sponge. You can see that is the asymmetric, and second one, see, you can see the radial symmetry, Echinoderms, and last is the arthropods and annelids. They have the bilateral symmetry. You can see that central one line, single ray that can divide this organisms into identical left and right half, only one plane. If you divide this vertically or horizontally, only if you can, only if you do divide vertically, no, there will not be similar half. But you can see here, wherever the line that passing through, any line that passes through the center can divide these organisms into two similar halves. But if you try to divide these organisms, sponges, you cannot make any identical half. That is called asymmetric. Next is the diploblastic or triploblastic organization. In the diploblastic organization, as the name indicates, animals in which the cells are arranged in the embryonic layers, there will be an external ectoderm and an internal endoderm, that is called as diploblastic animals. The animals in which cells are arranged in the embryonic layers, arranged in the embryonic layers, which an external ectoderm, that is external means outside, that is an ectoderm is there, and inner endoderms are called diploblastic animals. An undifferentiated layer called mesoglia is present between the ectoderm and endoderm. In between this ectoderm and endoderm, there is a undifferentiated layer called mesoglia. Example, porifera, Cnidaria. Next is a triploblastic. Triploblastic animals in which developing embryo has a third germinal layer called mesoderm in between the ecto and endoderm, and are called as triploblastic. There is another germinal layer, the third layer called mesoderm is present. Platyhelminthes to chordates. That is mean by diploblastic and triploblastic. Diploblastic, two layers will be there, embryonic layers, that is ectoderm and endoderm, and in between this ectoderm and endoderm there is an undifferentiated layer called mesoglia is present. Example, porifera and Cnidaria. And the triploblastic animals, the animals in which the developing embryo has a third germinal layer called mesoderm in between the ectoderm and endoderm, they are called as triploblastic. Platyhelminthes to chordates. Next is a coelom. What is my coelom? Coelom is a cavity, it's a body cavity that between the body wall and the gut wall, and it is lined by mesoderm. Okay, the body cavity between the body wall and the gut wall lined by the mesoderm is called as a coelom. Coelom simply itself called a body cavity. Okay, where it is present, this body cavity? Between the body wall and the gut wall. And what's the specifications or specific feature? It is lined by the mesoderm. And based on the presence or absence of this coelom, and animals that are classified in the acoelomates, coelomates and pseudocoelomates. The name indicates acoelomates, that is the animals in which the body cavity is absent. Example, platyhelminthes, they don't have any body cavity. So, those kind of animals that which do not possess the body cavity or the coelom is called as acoelomate. And coelomate, they are the animals that possess the coelom or the body cavity. Annelids, mollusks, arthropods, echinoderms, hemichordates and the chordates. These are the examples of the coelomates, that is animal having the coelom or the body cavity. Next is a pseudocoelomate. In the case of pseudocoelomate, the animal in that is in the pseudocoelomate, they are not all animals, but the some animals coelom is not lined by the mesoderms. That is, coelom is usually lined by the mesoderms, but in the case of pseudocoelomates, that is some animals that do not have the mesoderm that lines the body cavity. Instead, this mesoderm is present as a scattered pouches or rings in between the ectoderm and endoderm. Such a coelom is called a pseudocoelom. An animal that possesses the pseudocoelom are called as pseudocoelomates. Example is the Astyalminthus. Pseudocoelom means some animals coelom is not lined by the mesoderm. Instead, this mesoderm is present as a scattered pouches or rings between the ectoderm and endoderm. Such a coelom is called as the pseudocoelom. And the animal that possesses the pseudocoelom are called as pseudocoelomates. Example, Astyalminthus. So, these are the three types of animals that classify the animals into three types based on the presence or absence of the coelom. What is the coelom? The body cavity that is between the body wall and the gut, that is lined by the mesoderm, is called as a coelom. And the presence of coelom, animals which possess the body cavity or the coelom, is called as the coelomates. And animals which do not possess the body cavity are called as acoelomates. And in the case of pseudocoelomate, the coelom is not lined by the mesoderm. Instead, this mesoderm is present as a scattered pouches in between the ectoderm and endoderm. And such animals are called, such a coelom are called as pseudocoelom, and the animal that possesses pseudocoelom are called as pseudocoelomates. See the picture. You can have the picture or representation of the acoelomate, coelomate and the pseudocoelomate. Acoelomate, you can see that there is no coelom, and the pseudocoelomates, there is a pseudocoelom, there is a white line extra. See here, there is another ring, that is pseudocoelom. And coelom, see here is a body cavity between the gut wall and the body wall. Coelomates are annelids, mollusks, arthropods, echinoderms, chordates. Acoelomates, Platyhelminthes, and pseudocoelomate, Astyalminthus. And here see triploblastic, diploblastic, and in this you can see that coelom is there, white semi ring is there, right, on either side, that is called as coelom. And here this is a pseudocoelom, that is the white is fully covered and you can see there is a pouches, and this is a acoelomate, there is no coelom. Next is a segmentation. In some animals, the body is externally and internally divided into different compartments or the segments with a serial repetitions of at least some organs, not all organs, at least some organs they are repeating in every segments. That is, in some animals the body is externally and internally divided into segments with a serial repetition of at least some organs. In the case of earthworm, body shows metameric segmentation and the phenomenon is known as metamerism. In the case of earthworm, it is a metameric segmentation and this phenomenon is called as metamerism. Segmentation: in some animals, body is externally and internally divided into segments with a serial repetitions of at least some organs, and in the case of earthworm, they shows a metameric segmentation and is called metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed in the dorsal side during embryonic development in some animals. In some animals, there will be a notochord, that is mesodermally derived rod-like structure that is formed in the dorsal side during the embryonic developments. Animal that possesses the notochord is called as a chordates. Animal without notochord is called as a non-chordates. Example, Porifera to Echinodermata. That is, animal with a notochord are the chordates and animal without notochord are called as non-chordates. And what is a notochord? Is a mesodermally derived rod-like structure formed in the dorsal side during embryonic development in some animals. Next is the classification of animals. a general chart from the NCERT textbook. That is, you can see the kingdom is their animal kingdom, that's a multicellular organisms. Then the level of organization is there, symmetry is there, then body cavity is there, then the phylum are there. First one is a cellular level organization, then next is a tissue level, organ and organ system level of organization. In the cellular level of organization, mostly asymmetrical forms, okay, and they are acoelomate, they do not have coelom or body cavity. Example is a porifera. Next is the tissue level of organization or the organ system or the organ level of organization. Here they have radial symmetry as well as the bilateral symmetry. Those with the radial symmetry, they do not have the body cavity. Example, Coelenterata, Cnidaria and Ctenophora. And the bilateral symmetry, they have the coelom, they do not have the coelom, they have the false coelom. Those with the body cavity are called as a coelomate, and those without a body cavity are called as acoelomate, and those with a false coelom or not true coelom is called as a pseudocoelomates. And the acoelomates, they are the platyhelminthes, and the pseudocoelomates, they are the astehelminthes, and the coelomate, they are the Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. And in the case of Echinodermata, in the previous slides also we discussed, that is when discussing about the symmetry, Echinodermata exhibit radial as well as the bilateral symmetry at the different stages of life. Depending on their life stages, they actually shows the radial or the bilateral symmetry. These are about the basis of the classification of the animals, and is the first part. Go through this video. Thank you.